Welcome to today's nanotalk on viruses and vaccines. I'm Ophélie and this is Amélie.
Hello, everybody.
And we will be your hosts for the evening. As some of you might not be completely familiar with this event, let me give you a quick overview of what will happen. For every nanotalk, we invite two young scientists to give a 15-minute talk about their research field. After each presentation, you will have the opportunity to ask questions. The nanotalks have several aims. On one hand, the format offers a training platform for speakers to help them improve in the domain of science communication. It also facilitates networking between audience, speakers, and organizers. And on the other hand, of course, the audience gets to know a little more about current research and new developments. Tonight's program is especially interesting as it addresses important topics concerning all of us today. Now, Amélie is going to tell you more about Reach, who made this event possible.
Yes, thank you very much, Ophélie. And next, I shortly want to introduce Reach to you. Reach is an independent think tank for a science-friendly culture. The aim of Reach is to promote a dialogue between scientists and our whole society. And for that purpose, Reach organizes events that inform in an unbiased way about relevant topics in different areas, such as digitalization, health, and also environment. Reach consists of over 200 volunteers from different cities such as Zurich, Bern, Basel, and Lausanne. And if you're interested in Reach too, don't hesitate to sign up for our newsletter. The nanotalk this evening is part of the SIMPACT program. In this program of Reach, young people such as Ophélie and myself organize events. SIMPACT aims to make those who are interested in science also aware of their responsibility and to enable them to make a difference with their knowledge. Before diving into our topic of vaccines, I want to remind you that questions can be asked via Slido, and the code to ask questions is nano-april. We will have a first Q&A session after our first talk and a second Q&A session after our second talk this evening. And the first speaker I warmly welcome now is Dr. Steve Pascolo. He is Principal Investigator at the University Hospital of Zurich in the Department of Dermatology. This evening, Mr. Pascolo is going to talk about mRNA vaccines. During his intense research on this kind of vaccines, he even experimented with mRNA on himself. Thank you very much that you're here this evening, and I hand over to you, Mr. Pascolo.
Welcome to my nanotalk on mRNA vaccines. mRNA vaccines are not new. They are used for 100 years. The vaccines against measles, mumps, yellow fever, rubella all consist of mRNA viruses that are attenuated. When injected, these vaccines deliver their messenger RNA in the host cells. The host cells produce the proteins of the viruses and mount an immune response. What is new is a synthetic mRNA vaccine where the messenger RNA is produced by in vitro transcription and is encapsulated in a lipid bilayer consisting of 4 lipids. The mode of action of a synthetic mRNA vaccine is the same than the mode of action of a natural mRNA vaccine used since 100 years. mRNA is pictured here. This is a cell, a mammalian cell with a nucleus. In the nucleus, you have a DNA. Part of the DNA is transcribed into pre-messenger mRNA. In the nucleus, the pre-messenger mRNA is matured. It's capped at the 5' end with this structure added at the 5' end, and it's polyadenylated at the 3' end. This capped polyadenylated mature messenger RNA goes in the cytosol, where it's further modified and translated into a protein. This messenger RNA in the cytosol is transient. It's being there expressed in protein for a few minutes till a few hours. In vitro, it's possible to produce this mature messenger RNA. It comes in two flavors, either non-modified or modified. When it's non-modified, it's produced with the 4 canonical nucleotides ACGU. And this non-modified mRNA in the body is recognized by receptors in immune cells. These receptors, called Toll-like receptors, stimulate inflammation when they detect nucleic acids, for example, non-modified mRNA that is injected. So when you inject this non-modified mRNA, because of inflammation, you can do vaccination. And this is used and developed as anti-cancer vaccination, anti-allergy vaccination, and has been approved recently against COVID-19 as antivirus vaccination. Another way to produce mRNA is to modify the mRNA with pseudouridine. And this modification allows the mRNA to escape the recognition by Toll-like receptors. So this modified mRNA is not immunostimulating. You can inject it and do not trigger an immune response. That's why it's used for gene therapy. For example, for expression of therapeutic proteins like erythropoietin or antibodies, it can be used with modified RNA for cellular reprogramming or genome engineering, for example, with Cas9. Recently, in 2017, it was published that surprisingly, this modified non-immunogenic mRNA can also be used for vaccination when it's given intradermal or intramuscular. But so far, there is no evidence whether modified mRNA is better or less good than non-modified mRNA for vaccination. And both are used. This is how mRNA is produced in vitro. You need a few reagents. First, a plasmid. This is a DNA molecule made by molecular engineering and extracted from the bacteria. It's linearized with a restriction enzyme, and the linear plasmid DNA is transcribed using an RNA polymerase, nucleotides, and the cap analog at the 5' into mature messenger mRNA. After this transcription step, you add the DNAs to destroy the DNA strands, and then the mRNA is recovered by selective precipitation with lithium chloride or more advanced method to have higher grade of messenger mRNA. Each of these steps is robust, which means that linearization, transcription, DNA treatment always work, whatever is the starting plasmid, whatever is the sequence in the starting plasmid, whether it codes for a virus protein or for a cancer protein or for allergy protein. This process is always the same. It works always. Within a few hours, through the steps, you get mRNA. This is in the laboratory. In pharmaceutical production, it takes a bit more time because you need in-process control and end product controls on the mRNA. A few facts on synthetic mRNA. First, mRNA is very stable. Contrarily to what people think, mRNA is the most stable biological molecule. It's the only biological molecule that you can heat up to 95 degrees, lyophilize, freeze, and that doesn't lose its activity. If you heat up to 95 degrees DNA or proteins or viruses, they will be deactivated. It's not the case for mRNA. So mRNA is the most stable biological molecule. On the contrary, the formulation of RNA, the pharmaceutical formulation of RNA in liposome may not be stable. It may aggregate, it may change size, form over the time, or by freeze drying. So that's why the formulation of the mRNA vaccines have to be stored at a low temperature. It's because of the liposome, not because of the mRNA. RNA in liposome is already approved as a drug. It's called Onpattro. It was approved in 2018. It's a drug used to treat patients suffering from hereditary transthyretin-mediated amyloidosis. And these patients receive 30 milligrams of the RNA intravenous every 3 weeks. It's not a messenger RNA, it's a siRNA. So this treatment exists, and as you will see, the vaccine uses 30 micrograms, 1,000 times less RNA than in Onpattro. Fourth point, IVT mRNA vaccines are vegan. As I showed you on the previous slide, the production requires enzymes which come from bacteria and chemicals like nucleotides. So the production is vegan. Finally, the production of mRNA doesn't require big infrastructures. 1 million of doses of mRNA vaccine can be produced with 6 liters in vitro transcription. In comparison, when you produce viruses for vaccination, you need a few thousand liters of cell culture in order to extract enough viruses for 1 million of doses. A few words on the timelines of IVT synthetic mRNA vaccines. The first demonstration that in vitro transcribed mRNA can be used in vivo was published in 1990 by an American team who has shown in mice that in vitro transcribed mRNA coding for beta-galactosidase injected naked in the muscle is expressed into beta-galactosidase. 3 years later, a French team has shown that the mRNA coding for influenza protein and packaged in liposomes can vaccinate mice. This was the first disclosure of mRNA vaccine, and it was mRNA in liposome, very similar to the COVID vaccines that have been approved. Later on, some more teams have shown that naked mRNA or other form of packaging can be used to vaccinate mice. And my colleague Ingmar Her, who has done his work under the guidance of Professor Ramondse and Professor Gunther Jung, has published in 2000 this article, and we have created CureVac in order to develop the mRNA vaccines and bring it to the clinic. One of the goals of CureVac was, of course, to improve the mRNA vaccine and to produce mRNA for the clinic in so-called GMP production, good manufacturing practices. We could produce then mRNA to inject in people, and it was first injected into me to check that naked mRNA could also be taken up into the human skin. Here is my leg. I got a few injections of mRNA coding for luciferase, and a few hours after injection, under local anesthesia, we made punch biopsies here, and we measured in vitro whether the punch biopsies were expressing light, and that was the case. So these experiments, published in 2007, have shown that when you inject in human skin naked mRNA, you find the same result than in mouse skin. That means the mRNA is taken up and translated into protein, in this case, firefly protein. Now for the COVID-19 vaccines, the three big companies have been working since the beginning of 2020 on the vaccines, and they came up all with the mRNA vaccine in liposomes that is given intramuscular and that is coding for the spike of SARS-CoV-2. First to start was Moderna, who is using modified mRNA. The name Moderna means modified mRNA. They use only modified mRNA, and they have injected the first healthy volunteers on March 16. They had 3 doses in their dose escalation phase 1 study from 25 to 250 micrograms. BioNTech started 1 month later with the first injection on the 25th of April, and BioNTech had 4 different vaccines in the test in April, and it included non-modified as well as modified mRNA vaccines. And the dose range was from 1 to 100 microgram. CureVac has started later with the dose range between 2 microgram and 12 microgram of non-modified mRNA. The phase 1 studies are published, and they have shown that this formulation of mRNA in liposome at this different dosage is safe and well tolerated, although there were some side effects, which is expected for a strong vaccine, inducing a strong immune response. Importantly, all volunteers seroconverted, even at the very low dose of the vaccine. And they were neutralizing antibodies after the boost at the doses used for the phase 3 study. So, and for the registration, which is 100 microgram of mRNA for Moderna and 30 microgram of mRNA for BioNTech. CureVac is ongoing in its phase 3 study with 12 microgram of non-modified mRNA. The phase 3 studies from BioNTech and Moderna have shown 95% protection against COVID-19. And more recently in Israel, that have massively vaccinated its population, it was demonstrated that the BioNTech Pfizer vaccine gives a 94% protection against infection by SARS-CoV-2. Here is an example of the antibody response triggered by these vaccines. Here, the BioNTech Pfizer vaccine given in healthy volunteers. Some volunteers here got a placebo, and they don't have in their serum any neutralizing antibodies against SARS-CoV-2. These are sera from volunteers who have recovered from the COVID-19, and they have neutralizing antibodies against SARS-CoV-2. These here are the healthy volunteers who got the vaccine at different doses, 10 to 30 microgram, younger people or elderly people. You see that in all the cases at day 28, which is 1 week after the second injection, all volunteers have neutralizing antibodies against SARS-CoV-2, even at the low dose of 10 microgram, but even higher at the 30 microgram, whether they are young or whether they are old. So this shows how strong is the mRNA vaccine, even at this low dose in both young and elderly population. With that, I'm finished. You can find more detail about the mRNA vaccine in my recent review published in February 2021, and I'm looking forward for your questions.
Thank you very much, Mr. Pascolo, for your talk. We have already received several questions, and the first question we have received concerns the difference between modified and non-modified mRNA. And the question is, does modified or non-modified mRNA need an adjuvant for activating the immune system? And are modified and non-modified mRNA equally good in stimulating our immune system? We have already received several questions.
Do you hear me well? So whether the modification is needed or whether it increases or decreases the efficacy of mRNA, we don't know. It was never compared exactly non-modified and modified mRNA in vaccines. So BioNTech has done it. In April, they had exactly the same sequence of mRNA in the exact same formulation, in the exact same site of injection, modified versus non-modified. They didn't yet publish the data they had with a non-modified. They published the data they had with a modified, but not with a non-modified. So as soon as they will publish the data they had with a non-modified, we will see in humans the exact same sequence of mRNA in the exact same formulation, in the exact same site of injection, whether there is a difference in the side effects and in the efficacy. So far, there was no such precise exact comparison between modified and non-modified in vaccines. So both work, and non-modified has been used more, especially against cancer, than modified. But in the context of COVID, we don't know, and prophylactic vaccines, we don't know whether the modification is good or bad. In the non-modified mRNA, because it's not anymore a toll-like receptor agonist, it's highly probable, it was also kind of shown, that the adjuvant effect comes from the lipid, from the liposome itself, is inflammatory. So when you inject the non-modified mRNA in the liposome, the inflammation probably, highly probably, comes from the liposome, also from the damage made to the muscle by injection, but most probably, and data are supporting that, from the liposome itself.
Thank you very much. We already have several following questions. And one is, you mentioned that different companies have different dosages for the vaccines. And the question is, how does it come that the dosages are so different from company to company?
They're not that different. I mean, there are 100 microgram for Moderna to 12 microgram for CureVac. But if you remember the graphic I showed you from BioNTech, the vaccine worked with 10 microgram, with 20 microgram, and with 30 microgram. So especially in healthy younger volunteers. So BioNTech could have probably come also with a 10 microgram vaccine, it would have worked. So they choose 30 microgram, they could have chosen more or less. The point is that you want to have a dose that will work in really nearly 100% of the people. So even if a dose of 10 microgram from the BioNTech vaccine would have worked in all younger healthy people, you are not sure that somebody immunosuppressed or weak would respond. So companies have probably chosen a dose that is higher than the minimum dose. BioNTech would have probably worked with 30 microgram instead of 100, and Moderna would have worked probably, they would have used 30 microgram instead of 100. In their phase 1 study, the 25 microgram dose of Moderna was good. It induced immune response neutralizing antibodies in all participants, healthy volunteers. So, and again, BioNTech Pfizer also worked at 10 microgram in all healthy volunteers. So the doses was, all doses worked, basically, what they tested. So they choose a dose that they think was good, safe, and still would cover everyone, but they could have chosen different doses. So at the end, I think that all vaccines would work at 12 microgram, like the one of CureVac. So if Moderna or BioNTech Pfizer would be 12 microgram, they would also work. So they choose 30 microgram to be a little bit secure that there will be no one who will be resistant to the vaccine. And sorry, there is also small differences in the liposomes. It's not the same liposome in Moderna, BioNTech, and CureVac. So it's also possible that the liposome of one company is better than the other company. So it could make a small difference. And of course, there is a difference between the modified and the non-modified mRNA. So maybe the non-modified is better than the modified, but these are probably small aspects.
Before we go to our next question, there is a very short question which you could maybe answer, and it is whether the BioNTech Pfizer now is using modified or non-modified mRNA in the available vaccine.
The BioNTech Pfizer is using modified mRNA. They have tested non-modified and modified, and they have chosen modified for the phase 3, but we don't know why, because they didn't publish the results they had in phase 1 with the non-modified vaccine. But the registration for the BioNTech Pfizer is the modified version of the mRNA. For Moderna also is modified. And for CureVac, it's non-modified. It's not yet registered. We are hoping that they will get good results.
Another question some people asked is that you mentioned that it is actually quite easy to produce the mRNA vaccines, and now we have several delivery problems, and people are asking, why do we have all these delivery problems by the companies now if it is actually that easy to produce the mRNA vaccine?
What I've showed is that it's very easy to produce mRNA. So the mRNA is very easy and fast to produce. What is a little bit more demanding is the lipid bilayer around. So the formulation, this requires 4 lipids. The 4 lipids, the lipid chemistry is difficult to make lipids, to purify lipids is much more difficult than to make and purify mRNA. So the struggle is to get the 4 lipids that you need for the liposomes of Moderna, BioNTech, or CureVac, and to get the providers delivering you this lipid, and then to mix the mRNA with this lipid. It also requires some special installations. So the bottleneck is not the mRNA, it's the liposome. And that's why companies, Moderna, BioNTech, and also CureVac, are working on new nanoparticles, which will not rely on these liposomes, which will rely on other liposomes, easier to make with, for example, only 1 lipid, or on nanoparticles which require polyplexes, in polycations, for example, polymers, which are easier to produce in large amount than lipids. So the tendency now is to replace this liposomal formulation of mRNA by other formulations, because mRNA is easy to have. It's the liposomal formulation which is difficult to get and to make. And then it's the liposomal formulation which is unstable, which has to be stored at low temperature. So if you have a new formulation of mRNA, if you have naked mRNA or mRNA in an easy formulation, then it could be much easier to do and stable at room temperature. So the issue is really much more the lipid bilayer around the mRNA than the mRNA itself. The mRNA itself is easy to produce.
We have our next bit more general question, and this is whether all vaccines are preventing an infection with the virus, or do they just prevent the severe symptoms, or do they really prevent an outbreak of the disease?
The endpoints of the clinical studies were to prevent the disease, because you can measure the disease. So you can see in your placebo or in your healthy volunteers vaccinated, you can see how many have symptoms of the disease. So the endpoints of the clinical studies were really to see how many people suffer from COVID in the placebo versus vaccinated people. It's much harder to test the the infectivity, so under placebo or vaccinated. But now it came from the Israel data. So in Israel, that has covered this population, they could see the rate of infection, so non-symptomatic infection, just PCR positive people without symptoms. They could see how many people got PCR positive in the vaccinated people versus the non-vaccinated people, and they could confirm that there is 95% protection against infection. So not only against the disease it was shown in the clinical study from BioNTech, Pfizer, and Moderna, but now in Israel, we also know that there is 94% protection against the infection. Which still means, sorry, that there is 5 to 10% possibility when you are vaccinated to still get COVID and to still get infected. So people should not believe that there is 100% protection. So we hear sometimes people saying, yeah, I know someone who was vaccinated, he got COVID. Yes, it's 90 to 95% protection. So you still have people who get vaccinated twice with the BioNTech, Pfizer, or Moderna, and who still get signs of COVID, still get positive in PCR, because it's not 100%, it's 90 to 95%.
So it actually means that you still have to be quite careful, because it's not a really 100% protection.
Exactly.
Then we also have a question. So you mentioned that there are mRNA and vector vaccines, and we have a question whether there are also other vaccine techniques than vector and mRNA, and then what are the differences between the vector mRNA, and if there also exist other techniques?
Against COVID, there are 4 platforms that have been validated in the world: protein-based vaccine, mRNA-based vaccines, adenoviruses-based vaccine, and inactivated SARS-CoV-2-based vaccines. Inactivated SARS-CoV-2 has been mostly done in China, and we don't have it here in Switzerland. Protein-based vaccines is the Novavax. It has been approved in several countries, not yet in Switzerland, although Switzerland has reserved it, and it looks good, the data of protection of the Novavax vaccine, protein-based. The adenovirus-based vaccine, you know, Johnson & Johnson, AstraZeneca, Sputnik V. So the AstraZeneca has been reserved in Switzerland. It's not yet approved, but it's adenoviruses. So yes, there are these 5 different platforms: protein, adeno, mRNA, inactivated virus, 4 platforms that have all been used in the world, all been validated, being quite good. I must say that the efficacy data are best with the mRNA vaccines that protect with 95%, that provide 95% protection. Other vaccines like the AstraZeneca provide less protection. And also the mRNA vaccines, the 2, the Moderna and the BioNTech, Pfizer, protect against all variants, while the AstraZeneca, the adenovirus, the recombinant adenovirus, does not protect against the South African variant. So the mRNA vaccine seems to be giving the better protection and the broader protection.
We already have a next question concerning the current development of mRNA vaccine. And the question is whether there are technologies under development that reduce the large amounts of injected mRNA, which is necessary for a potent immune response.
What do you mean? So higher amount, or?
The question is probably whether there are technologies with which you can reduce the amount of mRNA that is needed.
All companies are working in getting immune response with less mRNA, because of course, if you take the case of Moderna with 100 micrograms per dose, if they manage to have a new formulation, the lipid or the liposome or the nanoparticle, that allows you to get 10 times better efficacy, then that means you need only 10 micrograms in your vaccine, which means with your stock of vaccine, with 1 kilogram, for example, instead of doing 1 million doses, you will do 10 million doses. So all companies are working in improving the mRNA vaccine. In the last 20 years, we have improved it so much. You know, 20 years ago, in the first clinical studies, we injected a few hundred micrograms every week to patients to get a good immune response. Now we inject twice 30 micrograms. So in 20 years, it has progress enormously, and with science it will further progress. In 2 years probably we will have mRNA vaccines with only 1 or 2 micrograms in it. In 10 years maybe with 0.1 microgram. So the improvement of the mRNA, of the formulation of the mRNA, will allow to continue to improve the mRNA vaccine, which means to make it even more efficacious, which means that you will get even lower dose to have a high immune response. That's constantly ongoing. It's not the end of the mRNA vaccine, it's the beginning, and it will be constantly improved which will mean reducing the dose that is needed in order to get a high immune response.
Thank you very much. We have another question which concerns whether you're going to be positive in a PCR test shortly after your vaccination, or is it the case that the test targets other gene sequences?
The test targets few gene sequences in the SARS-CoV-2, so you will not get positive if you get injected the mRNA coding for the spike. Also, it will stay anyway in the locally in your muscle of the arm, so it will not go in the nose. So no, you will not get PCR positive after getting injection of the mRNA vaccines.
Yeah. This sounds great. And maybe for our last question, maybe you can tell us whether there is already information on how the synthetic mRNA can be used as a cancer treatment.
It has been the focus of CureVac at the beginning and also of BioNTech to do mRNA vaccines against cancer. We know that it's hard to make vaccines against cancer on any platform, peptide, proteins. It's the same for HIV. So there have been a lot of vaccines that have triggered immune response against cancer or against HIV. But clinically, although you get an immune response, you don't get a control of the disease, HIV or cancer. So the mRNA vaccine have fallen under this data. So they induced the mRNA vaccines against cancer that have been tested have induced immunity against cancer. So that was successful. But this immune response, like in cases of other vaccines, did not efficiently control cancer growth. So now the mRNA vaccines are being developed to target mutations, and the flexibility of the production of the mRNA vaccine allows to have individualized vaccines. So what is ongoing now mostly is to sequence the tumors of patients, of cancer patients. The sequence, once it's known, you find the mutations in the tumor versus healthy tissues, and you can generate mRNA coding for this mutation and vaccinate each individual with its own mRNA. It's fully individualized that codes for its mutation. BioNTech does that with Genentech. They're in phase 2. So each patient receives its own mRNA vaccines coding for its own mutation. And from the biopsy till the mRNA vaccine is ready for one person, it's 3 months. So the flexibility of the production of mRNA allows this technology, which is new, to really target in a totally individualized way the mutation in each cancer. So in theory, it can treat any type of cancer, melanoma, lung cancer, etc., because you target the mutation that each patient has in its own cancer. So this is one development of the mRNA vaccine. It's allowed by the flexibility of the production of the mRNA vaccine, as we have seen in the pandemic. It takes only a few weeks to develop an mRNA vaccine. It's easy, and so it goes fast. So that's why cancer patients can get this new therapy. And this is combined also with some standard immunotherapy, radiotherapy, and chemotherapy to have a double effect on the cancer vaccination plus other type of therapies to synergize and really get rid of the cancer and prevent relapses.
Yes, this sounds very promising if we can also use the mRNA for cancer treatment. And I thank you very much that you were here this evening, Mr. Pascolo, and answered our viewers' questions. And I hand now over to Ophelie for our second talk.
Thank you for the
very much,
invitation.
Mr.
Good
Pascolo.
evening. Thank you.
Thank you so much for sharing your research with us tonight, Mr. Pascolo. Now, please let me introduce all of you to Dr. Ettore Moreira from the Friedrich Mischer Institute for Biomedical Research in Basel. Ettore has prepared a very interesting talk for us this evening. The title goes, "What will the next pandemic be?" Ettore will tell us about different viruses and explain to us what could cause them to become the source of the next pandemic. I would like to remind you to ask lots of questions on sly.do with the code nano-april. Ettore will then answer them after his talk. Thank you so much for being here with us, Ettore, and the floor is yours.
So hello, my name is Ettore. I am a virologist and I'm working at the Friedrich Mischer Institute, FMI in Basel, Switzerland. And I'm really happy that today we're going to talk about such an important topic that's pandemics. So the title of the presentation is Viruses, What Will the Next Pandemic Be? So So what you can learn from this presentation, I hope that at the end of this presentation, you're going to be able to understand what a pandemic is. You're going to discover important pandemics, learn why some viruses they have a greater pandemic potential than others, discover which viruses could be the next pandemic. And at the end of this presentation, I'm going to quickly talk about how to avoid and be ready for the next pandemic. So what is a pandemic? A pandemic is a disease occurring worldwide, cross international boundaries, and usually affecting a large number of people. A pandemic can be a new emerging infectious disease, meaning a new disease, or a disease that already exists, but is rapidly spreading. The concept of pandemic is intrinsically associated with immunity and how contagious a disease is. But I'm going to talk about it in the next slides. And you might have heard about the Spanish and Hong Kong flu, Ebola, Zika, MERS, and SARS coronavirus. They are all examples of viruses that already caused pandemics. In talking about it, so I just want to tell in the next slide about the history of some pandemics of this millennium. And in 2002, 2003, the SARS coronavirus 1 infected around 8,000 people and killed around 700 people. So almost 10% of the affected people died because of SARS coronavirus 1. Then in 2009 and 10, we had the swine flu with more than 200,000 people dying because of this influenza. We had in 2014-16 a pandemic caused by Ebola with around 30,000 people infected and more than 11,000 people dying because of this Ebola, affected many countries in Africa. We had in 2015-16 the Zika virus pandemic with 1.5 million cases of Zika virus. Interestingly, Zika doesn't usually kill the infected person, but it can cause a chronic disease. And now we are facing, the biggest pandemic of this millennium. That's the SARS coronavirus 2 that I checked recently, and more than 3 million people already died because of this disease. So what is the pandemic potential virus, and why some viruses have greater pandemic potential than others? So as I said before, this is associated with population immunity, because when a population, people don't have a previous immunity against a virus, then this virus can infect, in theory, everybody in the population that is susceptible. And usually this happens with new viruses, or if a virus already exists, this virus can mutate or rearrange in order to escape the immunity. And this happens usually with the viruses that have high mutation rates, and mainly with RNA virus, because RNA mutates faster than DNA. Also, viruses, they have pandemic potential when they are highly contagious, when they can be transmitted easily from an infected person to other people that get in touch with this person. This typical example of this is like when a virus is transmitted by air, for example. And zoonotic viruses are usually the ones that cause pandemics, because they meaning that a virus comes from an animal where it is adapted to infect these animals, and it can jump to a human and then transmit from human to human eventually. And this happens because if it's adapted to animals, it has mutations for that, that makes it adapted to animals. And once it jumps to humans, usually we don't have any immunity against this virus because it's a new virus. Then the virus can spread. So we have to keep an eye on all viruses transmitted by animals, like transmitted by monkeys, transmitted by wild animals like bats, or they have mosquitoes as a vector. There is a list of priority diseases, or diseases that need our attention because they can be the next pandemic. And as a virologist, I have selected 5 viruses that could be the next pandemic based on what they are causing recently and their genetic potential of being a pandemic virus. So maybe the influenza is going to be the next pandemic. as I said, influenza already caused, pandemics before, the most emblematic one being the 1918 Spanish flu. And we might be, or you might be familiar with the symptoms that are caused by influenza that are, muscle pain, or body aches, but coughing and sneezing being maybe the most typical ones. And influenza, when it's not the seasonal, but like the pandemic one, it can have really high fatality rates, up to like 50%. Influenza, as I said, is a zoonotic virus. It's transmitted usually by birds or pigs to humans. It has, the natural host being aquatic birds. And recently also bats were reported to have influenza viruses, but we still don't know about the pandemic potential of these bat influenza viruses. Influenza is an RNA virus, so has high mutation rates and also has more than one segment. It has actually 8 segments that can mix or reassort to produce new viruses that we don't have immunity for. It's highly transmissible because it's transmitted by air, by air droplets. And even though we do have vaccines against influenza, but they are against the seasonal influenza virus, we need a vaccine that could prevent against a new virus or like a pandemic virus. So we need a research for the development of a universal vaccine. There are several antiviral therapeutics, some being more efficient to some strains than others. Influenza actually affects so many countries that it's difficult to predict which country could be the one that could originate this pandemic virus. It could be in Asia, Europe, or America. But maybe the next pandemic is an Ebola virus. Ebola caused, a severe disease. Maybe the most typical symptom being the gastrointestinal symptoms that are caused by Ebola. This like bleeding, diarrhea, and vomiting. It has really high fatality rates, usually over 50%, being as high as 90%. Ebola is also a zoonotic virus transmitted from wild animals to humans, mainly from bats or monkeys to humans. Caused an outbreak, a pandemic in 2014-16, with many countries in Africa being affected and more than 30 people being infected with influenza, with Ebola virus. It's also highly contagious and can be transmitted by body fluids from infected person to susceptible people. It has 4 viruses, so a pandemic could be caused by any of these 4 Ebola viruses. And even though there is no available vaccine, there are 2 licensed Ebola vaccines that could be or have the potential to be used in case of pandemic, for example. Since the last pandemic, we also have an antibody treatment, antibody cocktail that could actually be used in people that are infected and they are progressing to a severe disease. If a pandemic would be caused by Ebola, it could originate mainly from Africa because many African countries reported to have the wild animals viruses. So it could come, for example, from Guinea, Liberia, Sierra Leone, and others. The third potential pandemic virus that I highlight here is the Nipah virus. Maybe you have heard or not, but Nipah is appearing more as a dangerous virus, mainly in Africa, in Asian countries. It causes, besides normal virus-related symptoms, the most characteristic for Nipah is encephalitis. It means inflammation of the brain. It has also a high fatality rate between 40 and 75%. Nipah is also a zoonotic virus transmitted from infected bats to humans or bats to pigs, then to humans. In India, in 2018, it caused 90% death in a small outbreak that we had in India. 19 people infected and 17 people died because of Nipah virus. It is transmitted by coughing because it's transmitted by secretions in saliva. And there is no treatment and no vaccine against Nipah, so it would be really difficult to control in case Nipah, transmits from human to human more easily. But I read and I checked that there is some clinical trials in progress against the Nipah virus. We need, to develop further. If Nipah would be the next pandemic, then it would most likely originate from Asia, because there's where we have recently cases being reported, mainly from Bangladesh, India, Malaysia, Singapore, for example. But maybe the next pandemic is caused by chikungunya. Chikungunya causes a disease that affects mainly the joints, causing joint pain and swelling, as you can see in the picture. What is interesting is that chikungunya usually doesn't kill an infected person, but it can cause a chronic disease. So the person infected by this disease and they have symptoms, this person might have complications for many months or even years that can compromise the daily tasks and even work. Chikungunya is also zoonotic disease. It's actually a droplet-borne virus because it's transmitted from monkeys to humans through a vector. There's a mosquito vector. In 2017 and 18, we had outbreaks of chikungunya, and there is no vaccine and no treatment for chikungunya virus. If a disease epidemic would be caused by chikungunya, it would most likely come from South America, like Brazil, reported yearly some cases, Africa, like Nigeria and Cameroon, Asia, Thailand, and India. And lastly, maybe the next pandemic is a yellow fever pandemic. Yellow fever causes muscle pain, headache, vomiting, but most emblematically cause bleeding in some organs and liver damage or liver failure. So that's why some patients have, yellow eyes. It has a really high fatality rates as well, between 20 and 50%. It's also zoonotic virus being transmitted from monkeys to humans, having mosquito as a vector. Brazil in 2016 reported over 2,000 cases of Zika yellow fever virus. And even though there is a vaccine, this vaccine is quite efficient against the yellow fever, this vaccine is not available everywhere. So we would need to make this vaccine production ready for the next pandemic. And there is no treatment for yellow fever. So once a person is infected, the treatment is usually just based on the symptoms. If yellow fever would be the next pandemic, it would originate most likely from South America in countries like Brazil, Africa like Kenya, Uganda, in Asia from India and Thailand. So how can we be ready and avoid the next pandemic? So we should avoid deforestation, because destroying the forest make the wild animals be in closer contact to cities and to humans, and be able to transmit the virus and other diseases. We should stop wildlife trafficking, because besides that being a crime, it also makes the closer contact between the wild animals and humans. We should fight poverty, because poverty is a way that in a way helps transmission of disease, because of the conditions, lack of hygiene, the sanitary conditions as well, proliferation of some vectors like rats and some mosquitoes as well. We should promote hygiene, because then we can avoid being infected. Increase education, so we know how the viruses are transmitted and how to avoid it. And we should invest in science, because the scientists are the ones that are going to produce new vaccines and treatments against pandemic viruses. With that, I finish my presentation. I'd like to thank FMI, the place I work, my grant, the ERC, and thank the nanoTalk group and you for listening, and I'm open for questions. So thank you very much.
Thank you so much for your talk. Ettore. There are already so many questions coming in. I will start with a first question, a general one. You have told us a lot about viruses, and people are wondering if bacteria could also cause pandemics, and maybe what are the main differences in how we combat bacteria and viruses.
Yeah, so I highlighted viruses because the talk was more focused on viruses. But the biggest pandemic ever in history was actually caused by bacteria, was the Yersinia pestis that caused the plague. That's actually true. That killed more than 200 million people. So much more than the biggest pandemic caused by a virus that was a Spanish flu that killed around 50 million people. So yeah, bacteria can also cause pandemic. But luckily we have antibiotics, and antibiotics are usually a broad spectrum. So they can fight many different bacteria, many species of bacteria based on what they share in common. So it's different from virus. Like for virus, you have to have vaccines that are specific for this virus. While for bacteria, we have antibiotics that can fight many kinds of bacteria. So it's nowadays, I would say it's a bit more unlikely to have pandemics caused by virus like we had with Yersinia pestis for the plague, for example. But we have to be really aware that many bacteria now are being more resistant, or becoming more resistant to some antibiotics. And this is a huge problem now, for example, for some sexually transmitted disease, like gonorrhea and chlamydia. Some species or some variants of these species, they are becoming a bit more resistant, and then the antibiotics are not going to work against these bacteria anymore. And then we have to go to alternative treatments. Yeah, so they can also spread and cause kind of epidemics. But real pandemics, I think that viruses, they have the greatest potential.
I see. So resistant bacteria could be a big problem, but viruses are maybe likelier to end in a pandemic, or to be the one starting a pandemic. People are wondering how maybe viruses can mutate and not cause global pandemics like SARS-CoV-1 did.
Yes, we were lucky not to have a pandemic caused by SARS-CoV-1, because SARS-CoV-1, as I said, it affected around 8,000 people, so it was considered a pandemic already, but it was contained quicker because it kills around 10% of the infected people. So comparing the mortality rate of SARS-CoV-1 and SARS-CoV-2 is a huge difference. SARS-CoV-2 kills around 2 or 3 per 100, so it's 3%. Actually, even a bit less than that. Some say between 1 to 3%, while the SARS-CoV-1 killed around 10% of infected people. So because it was much more severe disease, it was, also people developed the symptoms earlier than SARS-CoV-2. We could actually track the infected people earlier and isolate these people. So that's how it didn't become a huge pandemic like we have the SARS-CoV-2. That we have, the incubation time for SARS-CoV-2 is a bit longer, so people don't have huge symptoms in the beginning, so they can spread the disease much easier. And also the mortality rate is a bit lower. Something also that I would like to comment is that some viruses, they even cause pandemic, like SARS-CoV-2 cause pandemic now. They can become non-pandemic after some point if they adapt, if they suffer some mutations that make them, like not a problem anymore. They're not going to kill people anymore. They're going to just cause some like mild symptoms. This has happened, for example, with the Spanish flu. There was H1N1. It caused pandemic and it devastated 50 million people. But after some point, it mutated and got more adapted to people. And now we are infected still with the Spanish flu, with H1N1, but this became the seasonal flu. So it doesn't cause a mortal pandemic anymore. It just causes, mild symptoms. And this can happen also with SARS-CoV-2.
Okay, that's very interesting. So actually the pandemics we are more afraid of, so to say, are not the very aggressive ones, because we can control them faster, and they're not also the ones that are really not dangerous because they're not dangerous. So it's something in the middle. Now, we're talking about a very important topic and very current topic, but there's another one, and that would be climate change. And people are wondering if climate change can have an impact on pandemics, and maybe that they can spread better, or that there are going to be more. Someone mentioned the melting of permafrost, maybe letting viruses out.
Yeah, I think this question is very interesting. It's really hard to answer because this is happening, like recently, I mean, the last century, I would say so, like where we can notice this climate change, change the climate to make it a bit more warm. And this has changed already the potential of some countries to also spread some disease that before they could not. Now we have, for example, USA and some European countries that they have now some vectors of some of the disease that I commented before. Some diseases are caused, for example, by a mosquito, transmitted by a mosquito. So they are virus transmitted by mosquito. And these mosquitoes now, because of the climate change, they can go to areas that before were too cold for them because they are usually from warm areas, So maybe now USA is reporting more arthropod-borne viruses, virus transmitted by arthropods, like mosquitoes and some ticks, so tick-borne disease, for example. So and therefore I can say that this virus that before they would be more epidemic and cause more symptoms in people from tropical areas. These viruses now they can spread also to subtropical areas because these areas are becoming warm as well. So the climate change is impacting the distribution of some vectors that can spread some disease that were before not able to be transmitted in these areas. Regarding ice melting of the ice, for example, this can indeed bring to life some bacteria for sure, because some bacteria they can transmit disease by themselves. For viruses, it's a more complicated situation. Viruses only live inside hosts. If we have, for example, viruses that are frozen in an area, they should be protected inside the host. Otherwise, for example, RNAs, they are not so stable. Even RNA viruses, they are not so stable to the point that they can survive for hundreds of years in ice. So I think it would be unlikely. You can maybe detect some RNA viruses in melted ice, but it would be unlikely they would be intact enough to actually transmit to people or contaminate people. So I think the melting of the ice would be more important for bacteria and maybe for some DNA viruses. Yes, and indeed we have a DNA virus, because I only said about RNA because I think they won't mutate fast and they are more problematic. But we had already a pandemic caused by DNA virus, was the smallpox. They also kill millions of people. So we have to be aware that DNA can also cause pandemics, and some DNA can appear from melted ice. But I think it's more unlikely than bacteria should cause pandemic.
Okay, thank you so much. It's really interesting all that you have to say. We just have so many questions, so we kind of have to go on so that everyone gets the answer they want. I wanted to ask, so we have, like you have named a few reasons why maybe we are now going to have more often pandemics. What are ways to prevent them? Where should the research go? And maybe what are the differences that you think are significant? We see that different countries make different decisions and they lead to different results.
Yeah, I mean, we had a great talk about mRNA vaccine, and this is a revolution because now we can make vaccines much quicker than before. In 3 to 4 months, you can already have a vaccine that can be tested in some clinical phases. And many people are skeptical about it because they say, wow, coronavirus vaccine came in less than 1 year, while vaccines usually take 5 years to be ready. This used to be true, but nowadays we have technologies that can make vaccines much faster. So this is a way that we can be ready for the next pandemic, is being prepared for produce vaccines faster. And also, for example, for influenza, I said, we have to have universal vaccines, because some viruses like influenza, they mutate so quickly that vaccine has to be produced every year, for a different strain. But there is a way to produce universal vaccines if we actually target really well conserved proteins in viruses. So this has to be a focus of research as well. So I think that's the way you can get prepared, invest in science. Yes, and also being educated about how viruses can be transmitted, because as I said, most of these pandemics are caused by zoonotic viruses. So avoiding contact with these animals is a way also to avoid the transmission from wild animals to humans. But once it's transmitted already, then you have to be ready with vaccines and therapies.
You talked about research. Is it also, does it also make sense to spend money on research on anticipating the next pandemic, or do you think that, can you hear me? Okay, it seems like we have a problem with the sound.
Okay, now it looks it's back.
Okay. All right. I was asking, is it worth it to spend money on research to anticipate the next pandemic, or do you think that this is something that is too random, so to say, to invest money in, and we should invest more money in like making vaccines or things like that?
Well, I mean, the answer for this question as a scientist is easy. We do have to invest money, and we can really prevent pandemics to happen. For example, for coronavirus, we had the SARS-CoV-1, and vaccines started to be developed, but they totally stopped, the production of vaccines, or actually more the testing of the vaccines stopped after the SARS-CoV-1 because it was gone quite quicker, because all the reasons as I said. But like we know, as I said, we have some potential viruses that can cause the next pandemic, like the Nipah. There is no vaccine yet. I mean, some vaccines are being made, but they are still in clinical trials. What if Nipah would come right now? We would not be prepared for it. So we can indeed be ready for pandemics if we invest in development of vaccines before the pandemic already comes. Because some viruses, they are conserved enough that if you produce a vaccine right now, we can be ready for the next pandemic. Some viruses, they mutate much faster, like influenza virus. So then you have to invest in a new type of vaccine that would be the universal vaccine. This is possible to be done, and we still are not ready. We still don't have it. So we have a large amount of area in science that can be invested to prevent new pandemics.
Okay, so your opinion is to invest in everything that we know, and then when a new plague comes, we have a place to start. That's what you're saying.
Yes, but you don't have to prepare specific for one virus as well. Yeah. Like you can also prepare for the technology. For example, the mRNA vaccine is a technology that can be used against many viruses, and because it's really, you can do it much faster. So invest in science in a way that you can also invest in technology per se, you can actually make things much faster. So yeah, that's actually my answer as well.
Okay, thank you very much. Thank you so much for your talk and for answering our questions. There really came lots of questions about different topics into this small topics. So thank you so much. This was really interesting. I wish you all a nice evening, and I'll let Amelie finish.
Also from my side, many thanks to our two speakers and also to all our viewers and your interesting questions we had this evening. I hope that you all enjoyed our talks and that you could also expand your knowledge on viruses and vaccines. Enjoy the rest of the evening, and we look forward to seeing you again in our May nanotalk, which will be on brainwaves during sleep, and it will take place on Thursday, the 27th of May, and then again at 7 p.m. Have a nice evening.